So thank you very much. It's a great pleasure to be here. I appreciate very much the invitation. So today, as you heard, I'm going to focus on one of my interests, which is pulmonary hypertension. I spoke the other day on myocardial infarction and cardiac regeneration, but there are some overlaps and some similarities. So I imagine many of you don't know much about this disease. So it's an important disease. It's not a common disease. But it is quite devastating when it occurs. This is an example from one of our newspapers a few years back in Toronto, but it's typical of the problem. This is a 14-year-old girl who liked to ride horses, and for 2 or 3 years she began to develop symptoms of fatigue and shortness of breath, though she looked otherwise well, and it was thought to be psychosomatic. She went to see specialists and nobody could find anything wrong. Again, there aren't many definitive signs of the disease that can be identified easily clinically. And finally, she began to have fainting spells, which actually also were thought to be psychosomatic, until she finally collapsed and could not be resuscitated, and was taken into one of the major children's hospitals in the city and diagnosed with pulmonary hypertension, but it was advanced and she basically could not be resuscitated and died in that admission. So just a few facts about pulmonary hypertension. It can occur in different forms. The pure form, primary, or now, as it's now called, idiopathic pulmonary hypertension, is a rare disease. Often it's genetic, as we'll discuss. So there's a mutation in a single family of genes. And the incidence is around 2 to 3 in a million. So it's quite low. However, more commonly, pulmonary arterial hypertension occurs associated with other diseases. And these are often autoimmune diseases like scleroderma. And this is a much more common scenario. So it's not that uncommon a disease. There is a strong female predominance, so it's a disease mainly of women, though men also are affected. And it afflicts all ages, particularly the young, so children can get this disease, but the peak really is in young adulthood. So this is a devastating disease because of the age group that it affects. And there's a tremendous unmet clinical need. Although there are treatments that have developed, particularly in the last 10 years, none of these treatments are particularly effective. They will slow down the progression. They can improve symptoms for a time. But in general, all the patients will progress. And without lung transplantation, which is available to only a minority of patients, they will die. So the prognosis and survival is around 3 to 5 years after diagnosis for idiopathic pulmonary arterial hypertension. This is not a good prognosis. So it really is an interesting disease from a scientific point of view, understanding what's wrong, but also from the point of view of trying to develop better ways to manage these patients and hopefully one day a cure. So we don't really know what's wrong with the lung in this condition. There's some ideas, but we don't really know. The lung is a very important organ, as you know, and you're probably aware that we breathe air in and out to oxygenate the blood, but what you're probably not thinking about is how vascular the organ is. It is the most vascular of all organs, and that's not surprising because obviously for the lung to oxygenate the blood has to come to the lung. And the other thing that you may not know is the lung is the only organ in the body that has to accept all of the cardiac output. So your heart and your brain receive only a portion of the blood that circulates in the body. The lung has to accept all of it. And it does so normally at very low pressures. So the pressures in the arteries of the lung are normally just a little bit above venous pressure. So the normal lung offers almost no resistance to blood flow. And that allows it to oxygenate the blood efficiently. And this is true at rest, as you are now. But also if you run a marathon, the same thing. The resistance doesn't go up. The pressures don't go up normally in the lung very much. And it's able to accept even big increases in cardiac output with no increase in pressure. So this is clearly abnormal in pulmonary hypertension, where the pressures are elevated even at rest. And although we don't understand all of the mechanisms, we'll talk a little bit about some of the ideas, we do agree that the problem in pulmonary hypertension is a loss or pruning of the microvasculature. And these are perfusion MR images of a normal lung and a pulmonary hypertension lung. And I apologize, they're not as beautiful as the images I saw yesterday at the fantastic imaging center that you have. But they do show how massively the distal vessels are reduced in size and number, and how abnormal this perfusion is. So one question, and one of the analogies really, and this is a good time to have this analogy because it's the tree in summer in the tree in winter, and nowadays in Canada and in Sweden the trees look more like this, but it's the loss of this distal vasculature which prunes the microcirculation and results in increased resistance. So we'll talk a little bit about why this happens, and I'll try to share with you some concepts, new concepts we have about that, and then talk about what that means in terms of trying to reverse this and ways that we can actually improve the pattern of blood flow in the lung. So this is the primary question that I think there's a lot of debate about, which is what is the root cause of pulmonary hypertension? And there's lots of different ideas out there, and on the surface these ideas look to be quite contradictory. So it has been suggested it's a degenerative process, and this is work from our group mainly and others that have also agreed with this, that endothelial cell injury and then loss of endothelial cells by apoptosis can result in degeneration and loss of functioning arterioles. I'll show you some data to support that. However, there's another idea out there which also is supported by good data, that this is a proliferative disease, that there's increased vascular cell, endothelial, and smooth muscle cell growth, which can obliterate, narrow the vessels and obliterate the vessels. So a very different concept, but again, good data to support that. And finally, although it doesn't get as much attention as it probably should, there's good evidence that inflammation is involved. So this is an inflammatory disease in our patients and in our animal models. And of course, inflammation can result in local tissue damage and can exacerbate this process and contribute to vascular remodeling and occlusion. Now, the thing I want to convince you of is these are not actually separate processes. These are probably they're not mutually exclusive. They're probably interrelated, and one process leads to the other. So I think I'll show you that these things actually fit together. So let's begin with apoptosis. The view that's emerging now is that this may be an initiating mechanism in pulmonary hypertension. And it's largely the endothelial cell that undergoes apoptosis. And this is either because of a genetic defect, so the pulmonary arterial hypertension gene is the bone morphogenetic protein receptor 2. And I'll show you some evidence that mutations in this receptor can predispose to apoptosis of endothelial cells. And also, it can occur because of strong environmental triggers. We've had several epidemics of pulmonary hypertension due to diet medications, anorexigen, the latest being the fen-phen a decade or two ago. HIV patients are now living longer with chronic inflammatory disorders, and they're developing pulmonary hypertension to a significant degree. Patients with congenital heart disease that have overloaded circulation, increased shear stress, will develop pulmonary hypertension that looks identical to the idiopathic form, and many other examples. So we've actually known for many years that endothelial cell damage and death is important in this disease. This is the monocrotaline rat model of pulmonary hypertension. It's a workhorse animal model. Monocrotaline is a plant alkaloid that, for reasons we still don't well understand, is toxic to endothelial cells. I don't know if you can see this very well with the lights, but this is the normal lung, and this is tunnel staining in an animal 3 days after receiving monocrotaline, and the green nuclei are visible scattered throughout the lung distal parenchyma, and this represents endothelial cells undergoing apoptosis. This can be quantified, and you see over the 21 days in which pulmonary hypertension is developing, there is progressive increase in endothelial cell apoptosis, and this correlates with an increase in right ventricular systolic pressure at day 21, shown here. Right ventricular systolic pressure will reflect the pulmonary artery pressure, so that's a convenient measure in the animal models. What's interesting, though, is that if you block the apoptosis using a non-selective caspase inhibitor, in this case ZASP, you can reduce the degree of pulmonary hypertension in the model. So this shows you there's a causal relationship between apoptosis and pulmonary hypertension. Now, some years ago, it's quite a long time ago, we began to look at angiogenic genes in pulmonary hypertension to see whether this might be therapeutic. And one of the first experiments we did was to do gene therapy in, the monocrotaline model. This compares, so here's your pulmonary hypertension. This compares the effect of an empty null vector versus the vector containing VEGF. And you can see a significant improvement in the medial area in this case, but also the pulmonary pressures. This should have been, there's a mistake on the slide, I apologize for that. But what was interesting is when you look at the distal small arterioles, so this is where the problem is, at these very small precapillary arterioles, you can see that with activated caspase immunostaining, that there is an apoptosis going on in these very small endothelial cells. You also realized that this blood vessel is made up really of only 2 or 3 endothelial cells, which are all undergoing apoptosis. So you can imagine what that might do to the function of that blood vessel when these cells disappear. There's a good chance that it might just regress and no longer be functional. And this is a similar sized blood vessel in an animal that had received VEGF, and much less apoptosis was visible in the lung of these animals. So VEGF is effective, and of course we don't know exactly how it's working, but one possibility is that it is an endothelial cell survival factor in addition to being a growth factor. But I think the really seminal report that really established the central role of apoptosis in this disease came from the Colorado group. This is work by Norbert Focal and Ruben Tudor, and it came for a very different reason. So while we were doing this these kind of experiments, the Colorado group was really focused on the endothelial cell proliferation, which I mentioned earlier. And they likened it to a neoplastic or cancer-like disease. So there was abnormal proliferation of cells, they were resistant to apoptosis, and they felt that this was the reason that blood vessels were being occluded. And so they decided to use an inhibitor of VEGF, which is the main growth factor for endothelial cells, and this was the Sugen compound, to try to block endothelial cell growth. And although they don't like to admit it now, the reason they did this was they thought it would be protective in the disease. And so this is the hypoxia model of pulmonary hypertension in the rat, so another model. And you can see that over 3 weeks of chronic hypoxia, this is now mean pulmonary pressure. There is an increase in mean pulmonary artery pressure, though it's not extremely marked. However, when they gave the Sugen compound to block VEGF, rather than preventing this increase, they actually had a paradoxical response. It got worse. So again, if you put this together with our VEGF experiment, we gave VEGF, made it better. Blocking VEGF makes it worse. It's clear that VEGF is playing a protective role here. But what they saw, which is really intriguing, was they saw that at early phases of the model, they saw increase in tunnel stain, in apoptosis by activated caspase-3 staining, as I showed you in the monocrotaline model. But they also found, somewhat later stages, that there was increase in proliferative cells. And these cells then grew within the blood vessels. It's not so easy to see this here, but they ended up obliterating and blocking blood vessels. So this is exactly what we think happens in patients. However, it seemed to begin with apoptosis then leading to reactive proliferation. And in fact, they proved that was the case because when they used the inhibitor of apoptosis, ZASP, to prevent apoptosis in the model, they completely blocked the increase in the pressure and the appearance of these proliferative changes and the remodeling changes. So they could completely abrogate the model, rescue the model, when they blocked apoptosis. So this really showed us that, in fact, apoptosis is the starting point. It's the trigger for pulmonary hypertension. And then apoptosis induces a number of subsequent reactions, which include appearance of these proliferative reactive vascular cells that are important in vascular remodeling. So a little complicated, but the two things are linked. So I think we can understand many of the pathological features in this disease through the lens of apoptosis being a critical initiating mechanism. So smooth muscle cell hypertrophy, well, that's well known to occur when there's endothelial dysfunction. If you damage the endothelial cells, then you allow the smooth muscle cells to grow more, so that will contribute to that lesion. Intimal hyperplasia, again, is a lesion that occurs, well-known lesion that occurs with endothelial damage and endothelial dysfunction, so we can understand that. And as I mentioned, this is the lesion that we didn't understand well. This is the plexiform lesion. This is found in many, but not all patients with pulmonary arterial hypertension. And it's a very abnormal lesion in which you have almost a glomeruloid growth of angioid-looking structures with endothelial cells and smooth muscle cells intermixed. It usually occurs after bifurcations and at regions of arterial occlusion. And again, we don't understand well the underlying mechanisms, but it appears that this may be in response to endothelial damage and subsequent abnormal proliferation of remaining endothelial cells. So I'd like to talk a little bit about the genetics because to get further insight into the mechanism, I think we need to look at how this gene that's been associated with the disease may be linked to the disease. And we've known now for a decade, well, we know for a long time there are families, so it's familial in a small number of patients with the disease. And so, of course, that led two groups to identify the gene. They identified the same gene that was involved in the disease, and that's, as I mentioned, the bone morphogenetic protein receptor 2. And for those of you that, well, first of all, so 6 to 12% of these cases are familial, these are loss of function mutations. In fact, there's probably about 200 mutations that have, separate mutations have been identified in this one gene, all of which seem to lead to loss of function. The gene is a member of the TGF-beta superfamily, TGF-beta receptor superfamily. So this is a very large family, very complex family, and it does many things. The other thing that's interesting about this gene is it's found in almost every cell in your body. Yet this loss of function mutation results in a disease that only affects blood vessels in the lung. So there's got to be some important clues as to why that happens. Now, we found out, actually this slide's a bit out of date, it's probably about 70% or 80% of patients with familial PH have been found to have mutations in the gene, and up to about 30% of patients that have sporadic non-familial PH have mutations. So a good about a third or more of patients that we see in clinic will have a mutation in this gene. No other gene really has been associated. The only other gene is another member of the same family, but no other genes have been associated. So it really is interesting why this gene is involved. So the first thing that was done to try to understand how this gene might be related to pulmonary hypertension was to look at its function in vascular cells. So here's an experiment in which smooth muscle cells were treated with BMP, in this case BMP7, and they were examined for growth and cell survival. And you can see an exon 5 is being used as a marker of apoptosis. And you can see that the control cells, of course, did not show apoptosis. Cells treated with BMP showed the green staining, which means they're undergoing apoptosis. And if you look at the summary data by flow cytometry, you can see about a 3-fold increase in cell death, apoptosis, when these cells were given BMP. And there was also a decrease, not surprisingly, in cell proliferation. So this led to the idea that this pathway, BMP and BMP receptor, were involved in growth regulation of smooth muscle cells. And if you had a loss of function mutation, then that would make the cells hyperproliferative and subject to greater vascular remodeling. So that could certainly contribute to one of the major lesions seen in pulmonary hypertension. However, no one, up until the time we did this experiment, had asked the same question in endothelial cells, which are very different cells. And we know from the TGF-beta superfamily that the effects of ligands of TGF-beta receptors are very much dependent on cell type and the actual context in which you do the experiment. So in endothelial cells, we dumped a lot of BMP on our endothelial cells, and no apoptosis occurred. And then we induced apoptosis, in this case using TNF-alpha, which is a good way to kill endothelial cells. And you can see a lot of bright green tunnel-positive endothelial cells. And then we added BMP to TNF, and we could see that we could actually block apoptosis. So here we're getting the exact opposite effect in endothelial cells than what we saw in smooth muscle cells. And you can see the summary data here, TNF increasing tunnel positivity, TNF and BMP markedly reducing the apoptosis. So this actually isn't so surprising that we get BMP doing a completely opposite thing in the endothelium versus smooth muscle cells. So in the smooth muscle cells, it appears to be inhibiting growth and inducing apoptosis. In the endothelial cells, it's acting as a survival pathway. But if you think about it, both things are relevant to pulmonary hypertension. So if you lose function of this, so if you have a mutation in this pathway, you're going to increase susceptibility to apoptosis in the endothelium, which we know is bad and maybe a trigger for pulmonary hypertension. You're also going to make the smooth muscle cells more reactive. So they're going to grow more and narrow the vessels more. So it's like a double whammy for pulmonary hypertension. So it makes sense in terms of how this might relate to pulmonary hypertension. Just to prove that it was actually what the receptor is doing in endothelial cells, we knocked down the BMPR2 receptor using silencing approaches. And this shows you that we are successful in decreasing protein expression. And again, under basal culture conditions, we showed that when we knock down BMPR2, we increase basal levels of apoptosis in endothelial cells. Again, confirming this pathway is a survival pathway for pulmonary hypertension. Okay, so the next thing to do, of course, is we now know the gene. We can knock out the gene, so let's look at the phenotype in mice. Well, you can't actually look at BMPR2 null mice because it's embryonic lethal. In fact, the embryos don't even implant, so they die at the earliest stages of gestation. So we have to look at the heterozygote mice, which should be a good model because in humans, the mutation is haploinsufficiency, so there's only one mutated copy in patient that has familial pulmonary hypertension. So that should be reproduced by heterozygosity in the knockout. So The unfortunate thing is that the knockout model doesn't provide any phenotype. So if you measure, and a number of groups have done this, if you measure the right ventricular systolic pressure in the wild type versus the heterozygote knockout mice, there's no pulmonary hypertension. So very unfortunate. Part of this is probably due to the fact that the mouse is a very lousy model of pulmonary hypertension. Mice are very resistant to this disease. It's much better modeled in rats and other species, but mice don't really develop it. So that may be part of it. They're not very susceptible from a genetic basis. However, if you give them another hit, in this case serotonin, which has been linked to clinical pulmonary arterial hypertension. In fact, this is the mechanism of anorexogen-induced pulmonary arterial hypertension. It increases it. These agents, these diet-suppressing pills, block serotonin reuptake by cells. They increase circulating levels of serotonin. And serotonin has been shown to induce pulmonary hypertension. In the wild-type mouse, again, you see very little. Again, these mice are quite resistant to pulmonary hypertension. However, in the BMPR2 knockout mouse, you get a significant increase in pulmonary pressures when you infuse serotonin over 2 or 3 weeks. Interestingly, more recently, James West has found some dominant negative, or quotes dominant negative mutations in BMPR2 in families, and has expressed those in transgenic mice. So these are mutations that give a higher penetrant kind of pulmonary hypertension. Normally, patients with the mutation, only 20% will develop the disease. So with this mutation, greater proportions of patients develop the disease. And he's found that overexpression in a conditional binary transgenic doxycycline responsive model results in spontaneous development of pulmonary hypertension, again shown by increase in right ventricular systolic pressure in the animals that are expressing the mutated BMPR2 under control of doxycycline, so in the presence of doxycycline to induce expression of this gene. And this is also associated with right ventricular modeling, showing an increase in ratio of the right ventricle weight to the left ventricle weight. Interestingly, again, in the mouse, and this may be a specific feature of the mouse, despite the fact they develop pulmonary hypertension, they don't actually show increased muscularization of the arteries in the lung. So again, the mouse is not a great model of pulmonary hypertension, but the interesting thing from this is that clearly this is not necessary. It tells us the muscularization, though it's often associated with pulmonary hypertension, it may not be necessary. And again, this fits with our ideas that it really is the function of the distal precapillary arteriole that's important, whether it's being adequately perfused or not so much narrowing by muscularization. More recently, the same group, James West, has expressed another deletional dominant negative BMPR2 mutation in the same kind of DOX conditional system, and shown that in this case he gets, again, spontaneous pulmonary hypertension, but in a small number of animals. Again, it's only a fraction, but I think the fact it occurs at all is interesting, is you get occlusive changes within the lumen of these small arterioles. And this is sometimes by cells that are positive for VWF, von Willebrand factor, so this should suggest these cells are endothelial cells, but sometimes it's cells, and this is the combined image here, showing the smooth muscle cell by actin staining with the luminal area being occluded by these endothelial-like cells. But in other cases, it's caused by cells which are smooth muscle-like and express actin. So the obliterative cells are actually smooth muscle origin. So it's not always consistent, but what's interesting is you're getting obliterative changes, and these are the kind of things that we think are important in a human disease. We've collaborated with James West with that particular mutation, and we're expressing it in binary transgenic mice driven by an endothelial specific reporter, in this case V-cadherin, which is expressed in endothelial cells. In this system, you breed the 2 transgenic mice, and the binary transgenic mice will have both the TET transactivator and the transgene, so they'll be the TET transactivator will be turned on by doxycycline, and the transgene will be expressed in the binary transgenic animals. So again, we're seeing a modest degree of spontaneous pulmonary hypertension in this model. And this is associated with increase in apoptosis of endothelial cells. So again, consistent with the view that perturbation in the BMPR system predisposes to endothelial cell apoptosis. And what's really dramatic is what happens to perfusion. So this is our answer for your imaging center. Again, with the lights, you don't see it as well as you should. I don't know if the light can be turned off on the screen. But this is under low power, an intact lobe of a non-binary transgenic normal lung. It's being perfused with a fluorescent microangiographic solution containing very small microspheres in low melting point agarose. So it basically gives you a wonderful angiogram of the lung microcirculation. And it's immunostained, so you can immunostain this with an antibody to smooth muscle actin, the red. So this is what the mouse lung looks like normally. And this is a binary transgenic animal that has pulmonary hypertension. And you can see that what really dramatic finding is the decrease in distal branching. So that distal microcirculation, particularly the level of the precapillary arteriole that surrounds the capillaries, is absent. And at higher power, I think you can see, you can resolve these little tufts of capillaries. This is the precapillary arteriole, and the capillaries surround the air sacs, and this is the normal pattern of perfusion. And in the BMPR2 mutant mouse, you can see the precapillary arterioles there, but there is a lack of efficient connection to the distal capillary bed. And we think this is where the resistance is being generated, and this is the primary abnormality in this model. But also we see other changes, and these are, I think, are relevant to the development of pulmonary hypertension. There's an increase in inflammatory cells, particularly macrophages, in the distal lung in these animals. And there's an increase in cellular proliferation. So this is two different non-binary transgenic controls. One has only the ROSA. This is now a non-specific, the ROSA 26 promoter, which is ubiquitous. So we're now expressing in all cells. So this is the non-binary ROSA 26 mouse. You see a normal appearance. The non-binary mouse with the BMPR2 mutation, we do get a bit of leakiness. So this animal is showing a little bit of change in terms of increased proliferation, but it's not significant. And then the binary transgenic, which is efficiently expressing the transgene under control of doxycycline. You see a lot more positive cells by Ki67 staining. Again, the summary data is here. So to really try to understand this better, we generated this mouse. And it's sort of interesting mouse. It's expressing an artificial transgene called the fast-inducible apoptosis gene. And that consists of three components. One is a membrane targeting region shown here. There's two repeats of the FK506 binding protein domain. So this is a protein domain that binds to FK506, which is used as a immunosuppressant for patients post-transplantation, but also binds analogs of that which have no immunosuppressive activity, but bind very avidly to this region here. And that's used for dimerization. So we can dimerize the construct because the working end is the fast death domain, which is activated by dimerization. So basically, we're using this as a switch to turn on and off. When we add a small molecule, which is an inert molecule, which has no other biological effect, except to result in dimerization of this construct. So when it gets turned on, then it activates the death-inducing signaling complex, caspase, and then apoptosis. And we drive this under the control of an endothelial-specific promoter using Tie2, so that we can induce apoptosis only in endothelial cells. And we can titrate it very carefully by choosing the dose and the timing of administration of the AP506, as well as, so the AP2187, as well as by choosing mice that have the right expression profile. So interestingly, it does work. So when we develop these mice, this is one line that we're studying, you can see that we get a dose-dependent increase in right ventricular systolic pressure, even over 1 week when we give the AP compound. And this is associated with remodeling the right ventricle, so right ventricular hypertrophy. So that's exactly what we want to see. So we're seeing pulmonary hypertension. And the interesting thing is we're also seeing some dramatic remodeling in the lung arteries. And this is not in every animal, again, but it's in the animals that have the most severe pulmonary hypertension. And this is an example here. This is the peripheral lung, H&E staining, and you can see small arterioles here, and you can see normally they should be 1 or 2 layers thick in terms of cells, and you see it's very, very cellular, and looks very disorganized as well. It's almost like infiltrating cells. And in another region here, you can see, again, a little bit smaller arterioles completely replaced by these cells. In fact, in this case, the lumen is obliterated. So these cells are remodeling and even obliterating the lumen of these blood vessels. And if you look at the appearance, this is human. This is a plexiform lesion from a human patient with pulmonary arterial hypertension. I think it's remarkable the similarity both in the appearance of the lesion as well as in the location to a human plexiform lesion compared to this lesion that occurred in our mouse. Now, it does look a bit different. This is obviously much more cellular. However, this lesion occurred in 1 week. And this lesion probably took years to develop. So you're looking at a very different time frame. But it's not hard to imagine this lesion could mature to look like this lesion in a little while. So what are these lesions? Well, if we again look for cell proliferation by Ki-67 immunostaining, this is low power looking at the lung. You can see there's hot spots throughout the lung related to arteries. So there's proliferative cells around the distal arterioles. And if we do high power, again looking at that same lesion I showed you, you can see that this is highly proliferative, that most of these cells are undergoing proliferation, almost neoplastic. So again, getting back to that cancer paradigm for pulmonary hypertension. But this is induced by endothelial cell apoptosis, because that's all we did with the animal. It's an animal where we can turn on apoptosis of endothelial cells. So I think it's telling us that in response to endothelial apoptosis here in the lung, you get this tremendous reaction around the blood vessels. What are the cells? Well, we've begun trying to characterize the nature of these cells, and this is just a few panels showing you different staining, but there are endothelial cells. CD34 is actually the best for microvascular endothelial cells in the mouse lung. CD34 is the best immunostain to use. And you can see there are vascular channels within this, so there's neovascularization going on, but most of the cells are not endothelial cells. Most of the cells are CD68 positive macrophages. So it appears that, again, inflammation is coming in here. The reaction that occurs in response to inducing endothelial cell apoptosis in this model results in an inflammatory reaction, an innate inflammatory reaction, bringing in macrophages, which are highly proliferative. So I think, obviously we're exploring this further and trying to understand better how that happens, but I think this kind of experiments seem to bridge some of the contradictions in humans. I think now we can understand that apoptosis may be the central feature, the initial feature, and then the inflammation and the cell proliferation may result as a reactive response to that. Interesting thing is this only occurs in the lung. I'm not showing the data, but we have some apoptosis, in fact quite a lot of apoptosis that we can identify in the kidney, for example, in the arterioles of the kidney, but we see no abnormal changes. There's no inflammation, there's no proliferation. So it appears that the lung responds to this in a very specific and unique manner, and that may explain why the disease is so unique to the lung. So this is how we're trying to explain and understand all of these phenomena. So we think that under normal conditions, we may have endothelial cell apoptosis occurring from time to time in response to environmental stress. But normally the lung is able to repair the endothelium by local endothelial cell proliferation or other mechanisms, and we don't develop disease. However, if we have the BMPR2 mutation, or if we have a overwhelming environmental stress, this will overcome the repair mechanisms, and a number of things will happen as a consequence of increased endothelial cell apoptosis in the lung. So endothelial dysfunction may occur, because we know that regenerating endothelium cells are quite dysfunctional, and that can contribute to pulmonary hypertension through vasomotor mechanisms. We know the same mutations in BMPR2 contribute to smooth muscle hyperplasia plus endothelial dysfunction, so that can be part of it. I've already explained to you that we believe that actual degeneration or dropout of very small vessels, and that's I think what we're seeing mainly in the BMPR2 mouse models, and that can lead to a disruption and a discontinuity at the level of the distal pulmonary microcirculation. And then it's the reactive phenomena, both inflammation and vascular cell proliferation. It's not just endothelial cell that occur in response to the endothelial apoptosis and injury. And this may be the mechanism by which we get these complex intimal and plexiform lesions, which we see in many of our patients with pulmonary hypertension. However, although we spend a lot of time focusing on these lesions because they're so dramatic, one has to remember they appear late in the disease, usually in humans, and they're not always there. So patients can die of pulmonary hypertension, and the pathologist can go through the lung carefully and won't find the lesions. So clearly other things are responsible. We really do think that this degenerative change may be a fundamental problem in the lungs of these patients. So how do we take this information and use this to design new therapy? Well, many of these subsequent processes are amenable to various treatments. So for example, calcium channel inhibitors inhibit the proliferation of muscle cells, and that's being looked at in terms of trying to block potassium channels, or enhance potassium channels and decrease smooth muscle cell proliferation. As well, there's a variety of receptor tyrosine kinase inhibitors which are being looked at. They're used in cancer. Gleevec, for instance, sorafenib, are now being studied in pulmonary hypertension as well, with the idea that we can prevent abnormal proliferation of cells with these tyrosine kinase inhibitors. So there's actual trials going on now with both these agents. And although there's a lot of interest in it, the jury's out as to whether this is going to actually work. But the other completely different pathway one could explore would be to try to enhance the repair mechanisms. So to say, okay, all this is largely secondary to the damage that occurs, and is a reaction to the loss of endothelial cells. If we could enhance the repair mechanisms, reduce the consequence of apoptosis, then maybe these things would go away and we'd be able to restore and regenerate a more normal pulmonary circulation. So that's the idea we've worked on for a number of years. I mean, as usual, we began working on this well before we had a lot of the data I showed you, but it does fit together fairly well. And I'm going to focus now on, as we talked a bit more detail earlier in the week, about various kinds of stem and progenitor cells, but for the purpose of this talk, I'm going to only talk about adult progenitor and stem cells and those that arise in the bone marrow. And this, of course, occurred because we have known for many years that the bone marrow is a rich source of stem cells. For 30 years, we've been doing bone marrow transplantation, a very successful procedure for many malignancies. And that's because in the bone marrow, there's a hematopoietic stem cell, which can give rise to all of the elements of blood. So you can ablate the bone marrow and you can transplant a healthy bone marrow into the patient and they will recover their blood. But what we've learned more recently is the hematopoietic stem cell actually comes from a precursor that's called a hemangioblast, and this is equivalent to the cell in the embryo that gives rise to both the blood and the blood vessel. And the hemangioblast gives rise not only to the hematopoietic stem cell, but also to an angioblast, which we call an endothelial progenitor cell. So, in other words, in the adult, we have cells which are involved in repair and regeneration of blood vessels, the EPCs, and these can produce mature endothelial cells and new blood vessels. And they can be identified by a variety of different ways, but unfortunately, there's very little agreement as to what markers on the cell surface actually identify an endothelial progenitor cell versus any other kind of cell that's in the mononuclear cell fraction. And one needs probably to use several markers, and this becomes very inefficient, so you're dealing with less than 0.05 or even less than that % of the initial population. So it becomes very difficult to isolate enough of these cells to even do experiments, let alone do clinical trials. So the other way that you can actually derive these progenitor populations from the marrow-derived or circulating mononuclear cells is to do a culture selection procedure. And this is what was originally done by Isner's group at Sahara more than a decade ago, is to take mononuclear cells and plate them on fibronectin, so it's a substrate that endothelial cells like, and in the presence of endothelial growth factors like VEGF and other growth factors. And then very rapidly, you find that a population of these cells will adhere and look very happy under these conditions. Many other cells will not. They'll stay floating and you can remove them. And these cells start to express endothelial cell markers, so they become endothelial cell-like, but it's important to recognize that they retain the mononuclear cell markers, so they're still CD14 positive, CD45 positive, so they're still mononuclear cells. They don't proliferate, and that's good from a translational point of view, so they're probably safe to put into patients. And they are highly angiogenic. So if you use these cells in vitro models, or you inject them into animals in hind limb ischemia or myocardial infarction models, for example, these cells are very efficient at inducing new blood vessel growth, very angiogenic. So we call them early growth endothelial progenitor cells, but as I say, the term may not be totally accurate. They're probably really better called modified or angiogenic mononuclear cells. Interesting, if you leave these in culture for longer, so typically by 2 weeks, you'll begin to see these clusters of cells that form, and these are very different looking cells. The other thing you'll notice is the early growth cells begin to die off. They wane. They don't stay in culture for very long. But these cells are highly proliferative, and very rapidly they will overgrow the dish, and you get this beautiful lawn of very uniform looking cells, which in all intents and purposes appear to be mature endothelial cells. And they express no mononuclear or leukocyte markers. They express all of the endothelial cell markers. And they are identical to endothelial cells, except for the fact that they're much more proliferative than a mature endothelial cell would be. So these cells are interesting. They may have a lot of interesting uses for bioengineering, for example. There's been much less done in terms of understanding their therapeutic potential when injected into animal models, and it's unclear whether they have the same angiogenic effects as these cells. So we'll be talking only about the early growth progenitor cells in these experiments. So going back to the original problem, so pulmonary hypertension, back to the monocrotaline model in the rat of pulmonary hypertension, we asked the question, if we inject these progenitor cells, can we treat the disease? So we injected cells labeled with a red fluorophore so we could identify the cells, and you can see they lodge at the precapillary level. You can imagine a blood vessel here, and the cell gets gets caught at the distal pulmonary arteriole, and you see at lower power these cells are scattered throughout the lung. I'm not sure how well you can see that because of the light. Again, this is fluorescent microangiography, identify the distal arteriole, and you can see after 1 week, many of these cells are retained in the lung, and they reside at this distal arteriole level surrounding the arteriole in an endothelial position. And here's an example of a slightly larger arteriole in which the entire surface of the vessel now has been replaced by these endothelial progenitor cells, which are expressing or showing this red fluorescence, and it looks like they've taken over and replaced the damaged endothelium, and this would be an example of transdifferentiation. But having said this, I think although this can be found, it's not very common to see this, and we believe that other mechanisms are probably much more important in the biological effect of these cells, including paracrine mechanisms, and even an immune modulatory mechanism we won't have really a chance to talk about. I want to show you a movie because it's cool. Movies are fun. And this is intravital microscopy, injecting fluorescently labeled cell into an animal, in this case a mouse, and you're visualizing that's the lung. Okay, that's the phase contrast image. These are the cells which are being injected into the lung, and you can see how they're lodging at these precapillary arterioles and piling up. The interesting thing here is that there seems to be preferential perfusion, so you're delivering the cells preferentially to certain arterioles, and they don't block the arteriole. We always thought they would, but there seems to be good flow around the cell, so they actually don't occlude the vessel. This has been our big concern in moving this to a clinical trial, that we would actually worsen the pulmonary hypertension transiently as we injected these cells and microembolized them in the pulmonary circulation. So, what happens biologically in the rat model? Well, here you see the pulmonary hypertension that's induced by monocrotaline again. A somatic cell as a control, a fibroblast, has no beneficial effect, not surprisingly. But the EPCs injected 3 days after monocrotaline, so a prevention model, completely prevent the pulmonary hypertension. And not only that, they also completely restore the normal appearance of the lung. This is, again, microangiography like I showed before, distal precapillary arterioles perfusing the alveolar capillaries. This is 3 weeks after monocrotaline, and you can see the marked changes that occur, irregularities, what look like skips or occlusions, but this pinching off, this distal loss of continuity with the capillary bed, and this is largely prevented in the animals that receive these cells 3 days after monocrotaline. So very effective in preventing pulmonary hypertension. However, that's not a clinical problem. The real clinical problem is treating established disease. So the patients come to us, they have severe disease. So we need to show that this kind of therapy can actually restore a more normal structure and function in the lung in this condition. So in this experiment, we wait 21 days until pulmonary hypertension is developed, and then we randomize the animals to receive either monocrotaline or EPCs, monocrotaline alone, sorry. The animals all get monocrotaline at day 0. This is just monocrotaline alone, or monocrotaline with EPCs. Now, this is the baseline, so day 21, the pressures are similar. These experiments are done in a blinded fashion, again, to increase their validity, but it results in some noise in the baseline, so slightly different. However, when we look at the animals that receive only monocrotaline, you see that over that 2-week period from day 21 to day 35, we get a progression, and you'll see many of these animals die in this period. They get very sick. Whereas the animals receiving EPCs don't show this progression, so that's good. But what's not so good is that they don't actually get better. So again, in a treatment model, what we want to see is that we can actually make a sick animal better. And there really isn't any difference between the animals at day 35 and day 21. They just haven't gotten worse. So to try to make this somewhat more effective, what we've done is gone with a combination of gene and cell therapy. So based on work I haven't had a chance to show you, we have shown previously that endothelial nitric oxide synthase, which is a gene expressed normally by the endothelium and by endothelial progenitor cells, is very effective in the monocrotaline model in not only preventing pulmonary hypertension, but also in reversing established disease. So we decided to see if we transferred this gene into our cells and therefore overexpressed eNOS in the cells at the site of the vascular pathology, would this have a greater biological effect? And we did this using an electroporation system in collaboration with a company called MaxCyte. So now this is the model here. So again, it's animals that receive monocrotaline day 0. We do baseline pressures at day 21. This is what I showed you before, the monocrotaline alone. animals. And now when we look at the animals that receive EPCs transfected with enos, you see that not only do we prevent progression, but now we have a significant reduction in the pressures. And now the animals at day 35 don't look that different from the control. So this is what we want to see, actual reversal of disease. And quite impressive. And this is actually the same in mortality. This is that 2-week interval. You see that the monoclonal alone animals progressively die until nearly all are gone. The animals receiving EPCs alone have an improvement in mortality, but still you see this progressive decline. Whereas the animals receiving enos transfected endothelial progenitor cells show some initial loss early on, as you might expect, but after a few days the animals appear better and they no longer are dying. So again, consistent with the fact that we really have done something fundamental to improve the underlying pathology. So this is the basis of the trial that we have started. So this is the pulmonary hypertension and cell therapy, or FACET trial, which is a safety trial. Its primary endpoint is to look at the tolerability of administration of cells to these patients. And these are patients that have pulmonary arterial hypertension that are refractory to standard therapy. And we're giving enos transfected cells, much as we did with the rat, based on that preclinical data. These are autologous endothelial cells, so derived from the patient themselves. And they're delivered for safety reasons. We're doing this in a bit of a complicated manner. We're injecting the cells using a Swan-Ganz catheter. So that means we can measure the distal pressures in the pulmonary artery while we're delivering cells, because we're delivering the cells through the pacing port into the right ventricle. It gives good mixing. But we can measure the pressures during the delivery procedure. This is important because injecting a whole lot of these cells into patients that have critical increase in their pulmonary vascular resistance could be harmful if you were to microembolize sufficiently the lung microcirculation to increase the pressures, and that's what we're looking for. So it's dose ranging, and we give the cells over 3 days, again, to minimize the number of cells we give at any one period of time. So the way we do this is we isolate the cells from the blood using apheresis. So it's a machine where the patient gets hooked up to the machine, much like a dialysis machine. And it's basically like a small rotor or a big rotor, which the cells are centrifuged in the machine, or the blood is centrifuged, and the mononuclear cells are skimmed off. So it creates different planes of cells and collected in a bag. And in this way we can collect large numbers, billions and billions of mononuclear cells very safely and very easily. The cells are then cultured in the way that I showed you before to produce the early growth cells, and we culture them for 5 days. We then lift them off and transfect them together in collaboration with the MaxCyte with their electroporation device, which has been designed for human trials in GMP way. And then we put the cells back into culture for another 24 hours so that we can get rid of any cells that have been damaged by electroporation and make sure that when we lift these cells off and produce our final product, we're going to have high viability, in our case more than 98% viable cells. And then we produce a suspension at 2.5 million cells per ml, which is then delivered to our patient. So again, this is a bit of a complex, and you write these things, you try to maximize safety, but it gets a little complicated. So we have 3 dose panels. The first panel is 7 million cells, which we felt was a reasonable number to be safe for patients, but not likely to be effective. And to make it safer, we divide it into 3 separate doses on 3 separate days, starting off with a test dose of 1 million cells, which should be trivial for a human patient, and then increasing to 3 million for the next 2 days for that total of 7 million. 3 patients per panel, so if all goes well, we graduate to panel 2, in which the 3 million becomes a test dose. We go up to 10 million, total being 23 million, and finally we want to get to 50 million with a test dose of 10 million and 2 doses of 20 million cells. So that's where we think we're going to get efficacy as we've seen in the animal models. It's a total of 3 per panel. It's 9 patients plus an extra 3 patients at the highest tolerated panel, so we can at least have 6 patients treated with the highest dose, so we get some sense for both safety as well as potential efficacy, and I'll show you some of the preliminary data. There's a lot of inclusion-exclusion criteria. I've only shown you a little bit here, but they have to be very severe pulmonary hypertension, class 3 to 4 symptoms. They have to have great limitation in their ability to do that 6-minute walk. They're all listed, if appropriate, for lung transplantation. And initially we excluded any evidence of intracardiac or extracardiac communication because of concerns of these genetically engineered cells crossing over into the systemic circulation. Some of these things actually causes a lot of problems. So we had a lot of trouble recruiting initially because we found out that nearly no patient met our inclusion and exclusion criteria, mainly because if you do contrast echo and sensitive measures to look for intracardiac communication in patients with severe PAH, over 70% have 1 or 2 bubbles crossing the septum because of the gross deformity of the left atrial, of the sort of right atrium and the atrial septum. So we had to go back to Health Canada and our ethics boards and amend the trial so that we would exclude overt intracardiac communication, but allow the patient that has trivial 1 or 2 bubbles, because that's the majority. And we also expanded recruitment to include patients with associated PAH with scleroderma, as well as repaired ASD, actually. It's a safety trial, so we have a bunch of safety endpoints. I mentioned the hemodynamic monitoring during the delivery is the major safety endpoint, but there's a number of others shown there, immunologic, embolic, and so forth. And we're doing a fair number of functional and hemodynamic endpoints later on to try to get an idea whether we're making these patients better. So very quickly, I'll show you, we've done 7 patients so far. So I'll show you some of the data. And this is the hemodynamic data during cell delivery. So again, this is the main safety endpoint. The concern is this is pulmonary vascular resistance, total pulmonary vascular resistance, and the blue is pre-delivery and the sort of purpley color is 30 minutes after cell delivery. So the concern is that if we when we give these cells, we might induce transiently an increase in pressures, increase in resistance. As you can see, we don't see that, so this is very good news. So in the 7 patients, there's been no hint that injecting these cells actually transiently worsens the hemodynamics in these patients. That's good news. But the unexpected finding was that over that 3-day period, not immediately, but over that 3-day period of cell delivery, we get a substantial decrease overall in the pulmonary vascular resistance. And we didn't anticipate this. It's over 400 dynes per centimeter per second per centimeter to -5. So almost a third reduction. So the question is, remember these cells are genetically engineered to overexpress endothelial nitric oxide synthase. Is it possible that they're producing nitric oxide here, and this is resulting in an improvement in hemodynamics over that 3-day period? So it's certainly possible that's the case. So how could we get more data to maybe establish that more firmly? Well, it so happens, just to give you some biochemistry here, but nitric oxide synthase in the endothelial cells, it produces the nitric oxide from L-arginine, as you probably all know. This diffuses to the smooth muscle cell and activates guanylate cyclase, which is the enzyme that it is the target, the receptor, if you will, and it converts guanylate GTP to cyclic GMP. And the cyclic GMP is a second messenger that causes the vasorelaxation, also the growth changes that nitric oxide produces. But we also know that cyclic GMP is broken down by another system. It's the phosphodiesterase, in this case phosphodiesterase 5. And so it's the balance between the activity of guanylate cyclase and the activity of phosphodiesterase 5 that determines how much signaling you're going to get. And you may be aware that in the last decade or so, this drug has become very popular, Viagra, and it's an inhibitor of phosphodiesterase 5. So it inhibits the breakdown of cyclic GMP, enhancing cyclic GMP levels, enhancing the effects of nitric oxide. And that's why men who have certain difficulties use this drug. But it's also been introduced in the treatment of pulmonary hypertension because enhancing the effects of nitric oxide also is beneficial in patients with pulmonary vascular disease. So it's become a routine therapy for PAH. Of course, we changed the name. It's now called Revatio instead of Viagra because we're using it for different, but it's the same drug. And we tripled the price. So this is how drug companies do business. And it turns out that actually this is before Revatio, so our patients were on sildenafil. A natural experiment, half our patients were on a phosphodiesterase inhibitor, half were not. So let's have a look at what happens. So the ones that don't take, that were not, and this is just standard treatment, we didn't put them on it, this is part of their standard therapy. The patients not on sildenafil had very little hemodynamic change when we gave the cells. It was the patients that were on sildenafil that had the most of the changes. So that's consistent with the view that, in fact, this change that we're seeing may be due to increased nitric oxide production. We've got 5 more patients to go, and it'll be interesting to see whether this continues to be the case. If so, I think it would be fairly good evidence that this is, in fact, a biological effect of the transgene. In fact, probably the first evidence in cardiovascular systems that we can have a biological effect in a gene transfer approach. I just want to show you in closing some of the functional data. So this is done at 1, 2, and 3 months. And we're looking at 6-minute walk distance. This is a standard functional measure in patients with pulmonary arterial hypertension. In fact, this has been the primary endpoint of all of the trials that have been done for the PH drugs. It's the FDA-driven primary endpoint you've got to meet for your agent to be accepted as a clinical therapy. And what it means is it's a distance that a patient will walk unencouraged over 6 minutes. And it's a fairly good, not perfect, but fairly good measure of functional status. So at baseline, our patients were quite sick. They could barely walk 390 minutes in 6 minutes. However, at 1 month after cell therapy, there was an increase to 450. It wasn't sustained. It gradually decreased, which isn't surprising after one treatment with these cells. Again, you've got to be careful, though. This is not a blinded trial. There's no control group. There's no randomization. So we can't be sure whether this is a real finding or just a placebo effect. I'm just going to show you this is the same data actually converted to the actual change in 6-minute walk. So the actual change is 60 meters. To put that into perspective, most of the clinically approved agents for pulmonary hypertension will increase 6-minute walk by about 20 to 40 meters. So it's in the same range, maybe a little bit more. But again, non-blinded, so we don't know about placebo effects. However, when we do the same thing just for fun and say, okay, what happens in the patients that were not on the phosphodiesterase inhibitor compared to those that were on sildenafil at the time of receiving therapy? Is there a difference? And interesting, there is a difference. There was trivial changes in 6-minute walk in the patients not taking phosphodiesterase inhibitor, and there were substantial changes and more sustained changes in those that were on the agent. And this is up to almost 100 meters, which is a huge increase. So again, it doesn't prove it, but I think it's suggestive that in fact we are getting benefit in these patients. And the interesting thing is, just like in the animal model I showed you, that benefit seems to be more related to, or as much related to, the genetic engineering with eNOS than it is the cells themselves. So again, confirming the importance of enhancing activity with this approach. Yeah, that's basically what I just said. Just to tell you, we're not the only people in the world doing this. There's either 1 or 2 other groups, but they're in China. But this was published actually in JACC a few years back using a much simpler approach. Basically the same cells were using the cultured mononuclear cells, 5-day of culture, no transfection. And they were doing it from a simple blood draw. So they gave all the cells they could manufacture, so the range was 4 to 22 million, so they couldn't produce as many cells as we're producing. But they did a study which was randomized, and they had 31 patients roughly equally distributed between a control and a experimental group. And even though they didn't do any enhancement procedure, they did show actually significant benefits, but modest benefits, in all the endpoints they measured: the 6-minute walk distance, the pulmonary pressures, the pulmonary resistance, and the cardiac output. All around 10 to 15% improvement, which was, however, as I mentioned, significant. So interesting. Again, it needs to be reproduced, and we would say that we should be able, if we did the same kind of trial with genetically engineered cells, we'd expect more benefit. So, just to summarize what I've told you, I think apoptosis is necessary and sufficient to induce pulmonary hypertension in animal models. So I think this confirms that apoptosis is probably the central trigger for pulmonary arterial hypertension, leading to microvascular degeneration. Also reactive events leading to cellular proliferation and inflammatory response, all of which together we think creates the phenotype we see in patients with pulmonary hypertension. Enhancing endothelial repair by EPC therapy may be an effective new treatment for PH. It appears to be relatively safe, but we would feel that these cells by themselves, for a reason, are not sufficiently active to reverse established disease. But a combination of cell and gene therapy, in our case using endothelial nitric oxide syntase, does seem to provide some benefit. So we're hoping to finish our safety study soon, so we can get going with an efficacy trial to really see whether this therapy does work in these patients. So I'd like to thank you and